Hello everyone, thank you for your time. My name is RaminMehrani and I'm a PhD student at Ohio University working under supervision of Dr. Sumit Sharma. And please accept my apology for presenting virtually in an in-person session. So in my today presentation, I'm going to talk about the study of dimerization of interferon regulatory factor 3 proteins and ligand binding to IRF3 using molecular dynamics simulation.
We know that protein-protein interaction play a very important role in many biological processes inside our body, from cell signaling to cell division, and it also has a very important role in drug development or drug industry. So here are a couple of examples of the well-known protein that you may know, the hemoglobin protein and the insulin protein. But protein that today I'm going to talk about may not be that well-known as the hemoglobin or insulin, but it does a very important job in our body, which makes it interesting for a researcher to study on. So to actually look at the role of IRF3 protein in our body, so when our body is invaded by the pathogen, so in this case dsRNA virus, so innate immune cell, who are the first line defender inside our body, will try to send a signal to other innate immune cell about this invasion. So when the virus enters to the cell, it will be recognized by the help of pattern recognition receptors, in this case tool-like receptors 3, which activated tool-like receptors 3 release a cascade of signal to activate the single chain of IRF3 by phosphorylating that. So the phosphorylated IRF3 get dimerized and translocate to the nucleus of the cell and release a signal of the interferon alpha and beta to inform other cells about this invasion. So as you can see, the dimerization of IRF3 in whole cycle is a very important step, and researchers showed that aberrant activity of this dimerization resulted in so many diseases such as diabetes, cancer, and autoimmune disease. If you look at crystal structure of the IRF3 protein, we will see that it is a protein with 427 residues. And in this presentation, we only look at the last two domains of the protein, which are the interferon association domain and the autoinhibitory domain, shown by the magenta color and the green color in this right crystal structure. But from now on, in the rest of presentation, I will show both of these domains with the green color. So there are two schools of thought on the effect of phosphorylation and dimerization of IRF3. So in the first view, Quinn et al. In 2003 showed that dimerization happened after phosphorylation, but phosphorylation of IRF3 will result in significant change in the crystal structure of the IRF3 by separating the autoinhibitory domain from the IAD domain. In the second view, Takasi et al. In 2003 suggested that the dimerization happened after phosphorylation, but phosphorylation of the IRF3 won't change that much the crystal structure. But if you look at the crystal structure of the IRF3, non-phosphorylated IRF3 on the PDB or protein data bank, we'll see that there is a dimer structure. Might be, they suggested that it might be the artifact of crystallization. What we wanted to see if the protein IRF3 protein has a tendency to dimerize in a non-phosphorylated state or not. To do so, we looked at the dimerization free energy of non-phosphorylated IRF3 with respect to the reaction coordinate as a distance between the center of mass of the two chains of the protein, as you can see in this animation. So we used the steered MD simulation to generate the initial configuration, and we used the umbrella sampling techniques applying on the reaction coordinate, which is the distance between the two proteins. And we are going to do further analysis by using metadynamic simulation to find the best conformation between the two proteins when the two proteins are close together. In the second part of this study, we are studying the binding affinity of the ligand molecule to the IRF3 monomer. The ligand were suggested by our experimental collaborator, and we wanted to see if we can use the Markov state analysis to sort the binding affinity of these molecules according to the experimental result or not. To generate the initial configuration for umbrella sampling calculation, we performed a steered MD simulation over two different independent paths. So we separated the two proteins to the distance of 70 angstrom, and we changed the relative information at the distance of 70 to explore two different paths, two independent different paths. So here in this figure, you can see we begin from the distance of 70 angstrom. So the y-axis is the distance between the two chains of protein, and the x-axis is the angle between the two proteins, which represented by these two vectors that connected the alanine 222 to valine 76 and the corresponding alanine 461 to the valine 315. And the theta between the angle between these two vectors represent the angle between the two proteins. So the x-axis is this angle. As you can see, two different paths when we brought the protein from the 70 angstrom closer with the speed of 1 angstrom per nanosecond, and the two proteins come together until the distance is 30 angstrom, and they are exploring two completely different paths that later we will use to generate 81 different window for umbrella sampling calculation. Here are the results of these free energy calculation over two reaction coordinates of the distance and the angle that has been introduced in the previous slide. So as you can see, the color bar on the distance of 70 angstrom are said to be the same over two different paths, since when the two chains of protein are separated, so we assume that the energy level are the same. So as you can see, in the path 1, the protein will come through this path until a global minimum at the corner of this figure that showed by this crystal structure. So we will see some dimerization happening at the distance of around 30 to 33 angstrom with the well depths of around 10 kBt. If you look at the path 2 also come to very close local minimum at the distance of 34, but it cannot explore this global minimum. So there are a couple of local minimum in the path 2. So overall, this free energy calculation show that there is a dimer structure or favorable dimer structure for non-phosphorylated IRF3, but as we can see, it is path dependent that what would be the final dimer structure, or there are a couple of local minimum in the second pass as well. So to validate our free energy calculation, so we performed an unbiased simulation by separating the two proteins at the distance of 54 angstrom and leave them to see if they can dimerize or not. As you can see, the distance reached to the global minimum free energy at the distance of 34 after around 60 nanoseconds, and it stays there for a very long time. As we observed, the result of umbrella sampling shows some dependency on the path, and to fully understand this behavior, we will perform a metadynamic simulation by with respect to two different reaction coordinates when the two chains of protein are close to each other to find the exact dimer's location. So to do so, we defined two sets of vectors shown by the green and magenta color, and we defined two planes. So the plane 1 is connected to alanine 221 to valine 76 to alanine 461. So these 3 points is creating the blue plane 1, and the plane 2 is connecting valine 350 to alanine 461 to valine 76. So this is plane 2. So by the help of these auxiliary vectors and the two red vectors, we can generate two planes and then calculate the angle shown by the theta between the two planes. Same plane has been generated generated and the angle between generated by the magenta vectors, and the angle between them are phi, as you can see. So phi and theta are the reaction coordinate of our metadynamic simulation that we are performing to see the result of the favorable dimer conformation. But unfortunately here our result didn't converge, so hopefully soon I will later we will show we have some very exciting result from this metadynamic simulation. In the second part of the presentation, we are looking at the binding affinity of the ligand bind to the IRF3 protein using the Markov state model. So the assumption of this part is that the ligand binding to the protein is following a Markov process by moving from one state to another state of the binding. So using a Markov state model will help us to explore these bounded and bounded states and the unbounded initial states and the transition between them. To study the Markov state model on IRF3 protein, we ran multiple independent trajectories for each ligand molecule suggested by our experimental collaborator, these 3 molecules. And each of these trajectories is 200 nanosecond long. Then we use the Markov state model, as I mentioned, to find the unbounded and bounded states by combining all these microstates together. Since we didn't have any experimental result for the case of binding to the IRF3, we begin with testing the Markov state model on the protein of GSK3, which is the glycogen synthase kinase 3, which we do have experimental result for them. So as you can see, our experimental result shows that the COP187 has a higher binding affinity with respect to COP1-214, and the COP214 has the higher binding affinity compared to the COP168. As you can see, our Markov state analysis result, which showing 3 binding state and the unbounded state on the very right hand side, and the number below each figure are the stationary probability belongs to each state. As you can see, our trend also shows the same behavior. So we have a 70% chance of binding to the exact location that the experimental exact binding pocket that has been reported in the experiment close to the cysteine 199, and the COP214 shows 20% binding to that location, and the COP168 shows the 12%. So not only predicting the exact location of the binding, but the order also perfectly matched with the experimental result. Now that we showed that the Markov state model is working for binding prediction, we tried 3 different molecules, the COP187, phenylmetimazole, and COP214 for binding to the IRF3 binding. As you can see, we didn't observe that much of good binding between the any of these molecules and the IRF3 protein, and more of the numbers belongs to the unbounded state that you can see on the right hand side. So phenylmetimazole, there is an experimental result that showed that phenylmetimazole is an inhibitor of the IRF3 protein, and we also observed 21% binding to this location. But still, this part required more experimental validation to confirm that the order of binding of these ligand molecules. In conclusion, our umbrella sampling free energy calculation showed that non-phosphorylated IRF3 has a favorable dimerized structure, and but this free energy calculation is path dependent. So we need to do further analysis using metadynamic calculation to find the exact dimer configuration of the IRF3 protein. In the second part, we showed that the Markov state analysis it can recognize the order of the binding affinity according to experimental result, but binding affinity, and we also find that the binding affinity of the IRF3 protein with respect to 3 molecule, which required further experimental validation to confirm this result. With this, I would like to thanks Dr. Mondal, who helped us a lot in the Markov state part, and our group, and the National Science Foundation for financial support of this work, and High Supercomputer and Exceed for the computational resources. And once again, thank you all for your time, and I hope to see you soon.